Welcome everybody to this week's episode of 17 Minutes of Science. I'm Janice Weeks. I'm a co-founder and chief global health officer of In Vivo Biosystems. And it's my great pleasure to have as our guest today, Professor Ann Hart from Brown University. And she will be speaking about using C. elegans to model neurodegenerative disease. Anne got her BS at Michigan State, then her PhD in neuroscience at UCLA, working on Drosophila. She then went for a postdoc at Mass General Hospital and started working on C. elegans, and then stayed at Mass General and at Harvard University as a faculty member for a number of years, and then moved to Brown University in Rhode Island in 2009, where she's now full professor in the neuroscience department. I first got to know Anne as a fellow instructor in the summer neuroscience courses at the Marine Biological Laboratory in Woods Hole. So it's a special treat for me to see Anne again today and have her tell us about her work. So I'm going to start my clock. We'll have 17 minutes. If I can find my clock. There we go. Right, and if you have questions, just pop them in the message box in Facebook and we'll try to get to them. So Anne, you established the utility of C. elegans for modeling this area of research by establishing the first explicit models of human neurodegenerative diseases in C. elegans. So what was it about C. elegans that made you think they would be an ideal model for this sort of research?
Hey, Janice. First of all, it's a real pleasure to talk to you and about this, and I really appreciate the invitation. In effect, when I first thought of using C. elegans to make models of neurodegenerative disease, at one level, of course, I was standing on the shoulder of giants who had done work in Drosophila and others who'd done work in C. elegans, showing that there's really deep conservation of genes and molecules and pathways and how cells work in all of the animals on the planet. So as a neuroscientist, just starting my lab, I went to a seminar about Huntington's disease, and the audience, I was like, well, I wonder if I could model this in worms. It seems logical that we should be able to model disease just like we try to understand basic questions about how cells communicate with each other in development or other processes. So the deep conservation and also my background studying developmental biology and watching conservation.
Great. Well, so in your research, you use forward and reverse genetic techniques to identify pathways that are critical for these diseases. Can you tell us a little bit more about that approach?
Yeah, so the question of using genetics, both forward and reverse, to dissect something isn't new to your listeners, but it's, when I started as a scientist, using genetics as a tool was a surprise to me, but it's incredibly powerful. I think of forward genetics as asking the animal what's important in a process. So I'm going to say, I'm interested in neurodegenerative disease. So we can use genetic screens to figure out, what genes and therefore which proteins are important. So when we make a neurodegenerative disease model, right, we can say the neurons are dying in this model. Let's do genetic screens and find other genes whose perturbation suppresses. And that tells us something about why why things are dying in the model. So forward genetics is a tool of discovery. Reverse genetics is mostly what we're doing to try and make the model. So the explosion of information in human genetics in the last, decade or two is just amazing. And so now we know the genetic changes, the variations in the human population that can drive disease. And the reverse genetics we mostly are doing is we're taking those patient alleles, those patient amino acid changes, the change in the gene, and looking in the worm gene and saying, oh, we can make that same change here in the worm gene. And then let's see what happens. And sometimes we get neurodegeneration almost very, that looks in many ways mechanistically similar to what's happening in the patients. So the reverse genetics is making the models. Forward genetics is trying to figure out what goes wrong.
Great. So your research now focuses in two areas. So obviously neurodegenerative disease, but also you look at sleep and fatigue. So why did you choose to study both those areas, and how do they tie together, if at all?
Oh, yeah. When people look at the website, this is a really common question. So initially, there are two different threads. I started my lab after doing the postdoc with Josh Kaplan, studying sensory signals and behavior and how animals respond to the animal around them. And that also means understanding how the nervous system is responding and the mechanisms underlying behavior. And sort of that's been a thread running through half of my lab, going from sensory systems to the circuits and neurons and genes and proteins involved in responding. And then we shifted over to discovering, to our surprise, that the worms sleep, and then trying to figure out those pathways. It was initially a very separate thread to study neurodegenerative diseases. They've never really merged with each other. At one point in the past, we tried to see if neurodegenerative disease models cause the animals to have a altered sleep, and at least for the diseases I'm working on, it didn't. And another point recently, we're now checking to see if some of our neurodegenerative disease models cause fatigue or premature fatigue or alter the animal's ability to respond to exercise. So far, nothing much, but it's logical that it might happen. But either way, I think they're both fascinating topics of research that we can get at basic mechanisms using, these little tiny invertebrate animals. So I love them both.
Yeah. And for people who may not know, what does worm sleep look like?
Oh, yeah, right. So humans, of course, when we sleep is tied to the daylight cycle here on planet Earth. So we sleep at night and we're awake during the day, at least for the average adult. The worms live in rotting fruit or in the ground. They're really not as tied to the light dark cycle directly as we are. And so worm sleep seems to happen, for example, at certain times during development. So they will sleep when they're shedding their cuticle and probably rearranging their nervous system and doing a lot of developmental changes. So they sleep at specific times in development. They also, like us, will have stress-induced sleep. So if you have a fever or you have some kind of damage, work by David Ray's and others' labs has shown that you get the worms also have a stress-induced sleep as well. So, and probably mechanisms of arousal like alertness, like PDF receptor, those are probably conserved across species as well. So worm sleep isn't tied to the night-day cycle, but there's a lot of similarities, and we can lean back and say, we already know from work from our lab and others that molecular mechanisms that regulate sleep are also conserved as well. So it looks different also. I'll say normally worms are crawling around. Sleeping worm basically stops and bends its head a little and stops eating. It's not really dramatic.
Mm-hmm. Well, yeah.
But it's easy to track.
Yes, easy to see.
Yeah.
Sometimes humans, we sit around with our eyes closed and no one's, especially in Zoom calls, no one really knows if we're asleep or awake. So going back to neurodegeneration, what would you consider the most important finding from your lab in C. elegans about neurodegenerative disease?
I guess, well, first of all, there was the initial thing was that you could actually model a neurodegenerative disease in C. elegans, and people were shocked by that in the day.
Mm-hmm.
I think there's a lot more understanding now among the entire community about the conservation of behaviors, molecular mechanisms across animals on the planet. Probably one of the exciting discoveries more recently in the field is just how accurate these models can be. So for example, recently we published a model where we did the equivalent of knocking in patient disease variants into the C. elegans SOD1 gene. And when we did that, first of all, we were delighted to see that the right neurons died. So first of all, in ALS patients who have SOD1, either the cholinergic motor neurons of the spinal cord or the motor cortex glutamatergic neurons degenerate. And we have glutamatergic and cholinergic neuron degeneration in the worms. And in the patients, the dopamine, serotonin, those neurons are fine. Those neurons are fine in the worm. So that level of reflection across the millennia of how perturbation of SOD1 affects one kind of neurons versus the other was, initially I was like, that sounds too good to be true. But it's true. So the amazing specificity we can get with these knock-in models, I think that's pretty important, and it suggests that now we can use these in combination with people's overexpression models and other models as well to figure out what's actually going on. Why do some kinds of neurons die and not others?
Yeah, and sort of following up on that topic, so you've worked with Drosophila, the fruit fly, and C. elegans. So what don't people understand, or what's an important message to get out to people about working on invertebrate models versus actually working on the vertebrates or humans?
Oh, yeah.
To study these diseases?
That's a good question. So I think for people who are just entering the field, like a message you want to, whether we're teaching or talking to family or friends, is to make them understand that the animals on the planet are pretty much alike. So worms and flies, they're doing things the same way as larger animals like us. And it's important even in the scientific community, as the invertebrates start working in different fields, I find there's always a skepticism that really, you can use worms and flies to study learning? Really? You could use them to study sleep? You could use them to study neurodegenerative disease. Every time you move into a new field with alcohol, there's addiction. There's enormous skepticism from people who are using vertebrate models or studying humans that they can learn anything from the invertebrates. But we're all really related on this planet. And so I think that's what people don't understand, how similar they are. And I'll just throw something out there to say, if worms and Drosophila are so inexpensive compared to vertebrate research, if you look for how much money the NIH has put into basic research in microorganisms and invertebrates versus vertebrates, we've gotten a lot of payout from those dollars invested in tiny animals. I don't want to say that we don't need, vertebrate research. Absolutely. In fact, the testing non-human primates, COVID virus, there's so many things, learning, vision, but I think people don't understand the power of the invertebrates properly.
Right. And using, say invertebrates for looking at circuits and signaling pathways as you've done. And then, you do it in a cheap, easy, genetically tractable organism, and then you can track it up through vertebrates and so on. Yeah, I've worked on invertebrates my entire career too, so I'm right with you.
Yeah,
on that.
Someone said I heard a student here at Brown University say once, what we do here is we want to crash test our ideas fast and let the ones that are wrong die early. Let's crash test ideas and find out things and discard ideas and prove them in the small invertebrate models, and then go up to the mice.
Yeah. So let's see. So we're running, how much time do we have here? 3 minutes. So what do you enjoy most? I know you've said a lot of great things about C. elegans. You love working on it. I love working on them. What all do you, I mean, if you were to give a pitch about why C. elegans is so wonderful, I know one thing for me is the community of people, you know, good relationships among people, not so competitive in a bad way as some fields. So what do you have to say about the whole C. elegans world?
Well, the C. elegans community, before mentoring was a thing in the C. elegans world. And before collaborative research was really a thing in a big way in a community, C. elegans has always been collaborative. So I do love that part of the worm community writ large, on beyond even C. elegans, and even in the invertebrates. I mean, there are things I miss about Drosophila. I mean, this, after you do your PhD in something and it always has a little bit of your heart, there are beautiful techniques and tools there that are just being developed. But you know, remember, the size of the C. elegans community is really small compared to the Drosophila community, which is really small compared to the vertebrate research community. So maybe there's a camaraderie of knowing each other well. What I love about worms, how fast I can answer questions and do science and genetics. I can freeze them, I thaw them, and they come back to life. You can freeze the Drosophila; they don't come back to life. So they both have major strengths, and occasionally there's a day where I wish I was working with flies, but no, by the end of the day, I'm back to loving my worms.
Great. Well, I think that's the end of our, oh wait, no. Yeah, that's pretty much the end of our prepared questions. So what's something you'd like to talk about that I didn't ask you?
Something I'd like to talk about, I didn't ask you. Interesting. It'd be fun to talk about if we have 1 minute.
Something like that. Yeah, you can talk fast.
Would be how much we miss, and not just miss, but how much not getting together during COVID times has changed things. Like you and I got to know each other at MBL, whereas this giant, scientists from all over the country come together, we talk, we bump up against it, we get ideas. And now I've just so much missed that in COVID times. Virtual is not quite the same. You too?
Oh yeah, and I'm guessing everybody, online with us right now has that same feeling. Yeah, it's just not the same being on Zoom. It's just hanging out together and talking in the hallways and going out for a beer or coffee. Yeah, I'm with you on that.
Yeah, but I mean, there's been a few synergies that useful things from COVID will take with us, like the ability to, we're all much more comfortable talking virtually online to people across the planet. And we've gotten great seminars here from people all over the planet. But it'll be nice to come back, to get back together and sit down and have, a cup of tea with you.
Yeah, absolutely. Well, we timed that pretty darn well because we have 10 seconds now. So great, Anne. It's been such a pleasure talking to you. Hope you stay well and everything keeps going well in your lab. And we looking forward to everything getting back to semi-normal soon. So thanks again for taking the time to talk to us on 17 Minutes of Science. And for all of you out there in Zoom land, we'll see you in a week for our next episode.
Okay, thank you very much.
You too.